Hello everyone. This is going to be kind of a longer video, and it's a video that I've been meaning to do for a little while because the topic of the origins of COVID-19 have been in the news recently. And it's a topic that people have been talking about for the past year. Mainly, did the virus come from a lab or did it come from um, just a regular zoonotic event where the virus jumped from an animal? Um, so, and the reason that it's getting a little more press lately is because, well-respected scientists such as David Relman and Richard Ebright have come out and said that they believe that the lab leak theory is not one that should be easily dismissed. And this is a theory that was sort of dismissed when the WHO put out a statement fairly early on in the pandemic saying that a lab it did not escape from a lab, and there were some problems with that commission, namely that one of the people on the commission kind of had a conflict of interest in that decision, mainly the guy being Peter Daszak, who is the head of Echo Alliance, who gets a lot of funding from the NIH to study coronaviruses. So a lab leak would be fairly embarrassing for him. So, anyways, they said that it didn't come from a lab leak. So now I think it's important to clarify um, what a lab leak means. So there's kind of three major theories about the origins of the coronavirus. One is that it is a virus that was engineered, genetically engineered in the lab. And in this case, people suspect the Wuhan Institute of Virology. And two, the other theory is that it is a essentially natural virus that is not genetically manipulated, that accidentally escaped from the lab. And the third theory is that it was a natural virus that was in one of the animals at the Wuhan wet market that infected a person and started the thing. So let's go through those three scenarios, and maybe just like we'll read through some of the statements from these scientists. So they say the same thing. This is an interview with David Relman from Stanford. How might SARS-CoV-2 have first infected humans? So he says we know very little. The virus's closest known relatives were discovered in bats in Yunnan province, yet the first known cases of COVID-19 were detected in Wuhan, about 1,000 miles away. So basically, the one of the problems that they're pointing out is that the closest known viruses are in bats that are in the Yunnan province, and that is far, far away from Wuhan. But the first known human cases of COVID-19 were in Wuhan. So if the bats are in Yunnan, why are the first cases in Wuhan? And one possible explanation is that, well, the Wuhan Institute of Virology is there and it escaped from that lab. Um, so there are two general scenarios by which this virus could have made the jump. First, the jump or spillover might have happened directly from an animal to a human by means of an encounter that took place, say, within a bat cave or at an animal market. This is the current prevailing theory. And just so you know, before we go on, like this is my opinion. No one knows the origins of this thing. Like no one knows it, unless it actually was engineered, and the person who engineered it knows it. But obviously, they're not going to talk about it. Um, but barring that, no one knows the result, and this is just my impression of based on the current state of information. If the new information comes out, obviously we should be able to re-examine our position on these things. But anyways, so one scenario is it made the jump from people, or it could have happened indirectly with some other animal as the primary host, presumably bat had like it went through a civet cat or a pangolin or something like that. That's one of the current theories. So bats and other potential hosts are known to be shipped across China, including Wuhan. But if there were infected animals near Wuhan, so we have not found the animal that has this virus or has a virus like almost exactly like it, which is what you would want to find. So that's the other problem. The other problem is we have not found the animal that basically had the virus, had the SARS-CoV-2 virus. So maybe someone became infected with the animal near Wuhan and then moved to Wuhan. Then, because of the high transmissibility of the virus, you found expected other people near the site of its initial encounter. So again, the theory is why didn't we see infections near Yunnan where the most similar viruses are found in bats naturally? And two, why haven't we found the animal that it came from? So those are the two kind of problems with the natural jump theory. So what's the other scenario? SARS-CoV-2 could have spent some time in a lab before encountering humans. We know that some of the largest collection of bat coronavirus and a vigorous research program involving the creation of chimeric bat coronavirus by integrating unfamiliar coronavirus genomic sequences into other known coronaviruses were located in downtown Wuhan. We know that laboratory accidents happen everywhere there are laboratories. All scientists need to acknowledge a simple fact. Humans are fallible and laboratory accidents happen. This is all true. So we know that the Wuhan Institute of Virology has, they were collecting coronaviruses from all over the place and studying them. And they had made chimeric bat coronavirus where they combined the genetic sequences of one coronavirus with another. And this is the quote gain of function research that people are talking about. And like this is one of the papers out of the Wuhan Institute of Virology. And this is the lead author is Dr. Shi, and she is one of the foremost researchers on coronaviruses. And basically, they're talking about a chimeric virus, and we'll get back to this paper um, later on when we talk about some of the problems with the lab leak theory as well. Okay, so this other thing is that they were studying coronaviruses, they were making recombinant viruses. It's possible to escape from there due to an accident. And then other things like why is it important to know, and things like that. Okay, so makes sense. And this is Richard Ebright, um, a microbiologist, I think at Rutgers. And so, he basically says the same thing. Um, Whenever asked if this virus could have leaked from the Wuhan, your answer has been yes. This cannot and should not be dismissed. And then there is another guy, Stephen Quay, who is not a microbiologist and not a molecular biologist. And he put out this paper talking about Bayesian analysis. His paper has a lot of problems, so I won't go into it. And he misunderstands a lot of the molecular biology of the thing. Whereas I think Ebright and Albert are very good and they understand the molecular biology probably way better than I do. Um, but Stephen Quay definitely does not understand the biology. I read his opinion piece in Wall Street Journal, and he is not understanding it correctly. He assigns this 99.8 probably. Anyway, um, Dr. Ebright states, at this point in time, there is no secure basis to assign relative probabilities to the natural accident hypothesis or the laboratory accident hypothesis. So he also says like this is bunk. You can't like assign probabilities. We just don't have the information to assign the probabilities. Um, and then I think the important thing here from this interview is, people are asking, okay, okay, so this is important. What biological evidence regarding the structure and behavior of SARS-CoV-2 points to a pure zoonosis? That means a natural jump from animals to people. So he says the genome sequence of the outbreak virus indicates that its progenitor was either the horseshoe bat coronavirus, RATG13, or a closely related bat virus. Okay, so this is the same one that Dr. Relman was talking about. This is the one that's found in the Yunnan province, and then it was partially sequenced by the Wuhan Institute of Virology, and this was published in 2020. Bat coronaviruses are present in nature in multiple parts of China. Therefore, the first human infection could have occurred as a natural accident with virus passing from bat to human, possibly through another animal. There is clear precedent for this. So this has definitely happened before. SARS, MERS, all kinds of stuff occurred this way. But bat coronaviruses are also collected and studied by laboratories in multiple parts of China, including the Wuhan Institute of Virology. Therefore, the first human infection could have occurred as a laboratory accident, with the virus accidentally infecting a field collection staffer or a laboratory staffer, followed by transmission from the staffer to the public. This has also happened in history before. There have some of the entries of SARS into the human population occurred as laboratory accidents. So this is not an unprecedented event either. So both of them are saying that they could have been laboratory accidents. What neither one of them are saying is that it was an engineered virus. Okay, and that's pretty clear. Um, let's see where he says this. Yeah. The lab leak hypothesis, as raised in the open letter, should address four possible scenarios that do not imply genetic manipulation in a gain of function experiment. But this chance cannot be excluded. Dr. Ebert, when the pandemic started, you declared there is absolutely nothing in the sequence of this virus that indicates the virus was engineered. Would you reassert this today? And he says, the genome sequence of this virus shows no signatures of human manipulation. So basically, what do we know about these human manipulation signatures? Well, it basically comes from this. So this is a paper, and I'll link this below, published in 2014, and it basically talks about coronavirus reverse genetic systems. All right, so abstract. Coronaviruses infect humans and many species, are associated with a lot of diseases, and the large size of the coronavirus genome and the inability of some replicase gene sequences during propagation, instability of these replicase gene sequences during propagation presents serious obstacles for the development of reverse genetic systems similar to those used for smaller viruses. So the problem is that some of the enzymes don't work in a bacterial system, and the genome is quite large. So this virus is not easy to manipulate. And because of that, they have developed three systems, and they were difficult to develop. And that's why they talk about this in this paper. Uh, and let's see, where in this paper are there? Okay, yeah, here we go. However, the large size of the genome and the instability of some replicase gene sequences and the difficulty to synthesize full-length transcripts in vitro have hampered the generation of full-length. So they're saying this is a very difficult problem, but recently these problems have overcome employing three creative non-traditional approaches based on the use of bacterial artificial chromosomes, in vitro ligation of cDNA fragments, and vaccinia viruses as vector propagation. So there are three systems. They were all published around the year 2000. And they took a lot of work to develop, and these are the systems that are being used. And if we go back to this paper published from the Wuhan Institute of Virology talking about these gain of function systems, they used the bacterial artificial chromosome technique. So that's one of the three techniques here. And what's important to know is that all three of these techniques have identifiable genetic fingerprints. So when you sequence the virus that comes from the use of any of these systems, there's a scaffolding left over, and they leave a fingerprint, and people can realize if one of these systems are being used. So what's clear is that SARS-CoV-2 does not have the fingerprints of any of these systems. So the fact that it's engineered is unlikely because of that, and some other reasons that we can go into. All right, so the major problems that people who like the lab leak theory, like to point out is one, we have not identified the animal. There has, as far as we know, no animal has been found that has the virus that basically is the ancestral virus for SARS-CoV-2. So that's problem number one. Problem number two, the initial human known infections were in Wuhan, which is 1,000 miles away from where the natural vectors are, and coincidentally happens to be right next to the virology lab where they actually study coronaviruses. So that's number two. Number three is that the the spike protein works very well against people, which makes people think, well, maybe this was bred or engineered to infect people well. And number three is there's this thing called a furin cleavage site um, that is not found in a lot of coronaviruses, and they're saying that it's so rare that this furin cleavage site had to have been manipulated and placed in there. So we'll try to address all of those concerns kind of one by one. All right, so in terms of um, was it genetically engineered? Well, number one, we'd have to use one of these three systems, or I guess there's a possibility that someone developed a system. So it's a very difficult to develop a system, but they developed a system, they did not publish it, and they used it to create this virus. Okay, so that is a possibility. Very unlikely because most scientists, when they do something difficult, they like to get some recognition for it, and so they will publish it. But it's possible, like maybe there was some super secret lab that was doing it and they didn't want it published, and they created a system that no one in the world knows about, and they use that system, which has a fingerprint, but is a fingerprint that no one would know to look for. So that's a possibility. So, but as far as we know, that's a kind of an unlikely possibility, and they would have had to use one of the three systems that we know about. And if they did, it doesn't have that fingerprint. So, doesn't have a known fingerprint. Number two, this paper goes over some of the other problems with the manipulation theory. So, it says that the mutations in the receptor binding domain of SARS-CoV-2. So what they find out is that the SARS-CoV-2 seems to have a receptor binding domain that binds with high affinity to ACE2 from humans, ferrets, cats, and other species. So it's not just people, it's other animals too, but it does bind the ACE2 um, of people very well. So while the analysis suggests that SARS-CoV-2 may bind humans with high affinity, computational analysis predicts that the interaction is ideal and the receptor binding sequence is different from those shown in SARS-CoV-1 to be optimal for receptor binding. So what does that mean? It basically means, let's say that you were engineering a virus to have high affinity to human ACE2. Uh, you would do it two ways generally. You would find a natural sequence that is known to bind human ACE2 and then try to maybe either use that directly because we know that it works, or try to tweak it a little bit so that it works better. So the one that we know that binds ACE2 is SARS-CoV-1. Well, they don't say one, but it's the first one, the first SARS, the one that was developed in 2000 and one that was discovered in 2002. So that one has a sequence that we know binds human ACE2, but this one has a sequence that's fairly different from that. So it obviously didn't come from that. Or someone would have used a sequence that our computer analysis would have predicted a high affinity. But when you plug in the sequence of SARS-CoV-2 into our computational analysis, it says that it wouldn't work very well. So why would someone have used a sequence that would have been predicted to not work well? It doesn't make sense. All right, now talk about the furin cleavage site. So people are saying that the furin cleavage sites are rare, and so they must have been put in. But um, so it says here, polybasic cleavage sites have not been observed in related lineage B beta coronaviruses. That's what SARS-CoV-2 is. It is a lineage B beta coronavirus, and it's not observed in other of that lineage. However, other beta coronaviruses, so other very similar viruses, including HKU1, have these sites and predicted O-linked glycans. So it's possible, it's likely that they get these sites. And in fact, there's a paper right here that says furin cleavage sites occur naturally in coronaviruses. So these sites are common in coronaviruses, and you can get them through recombination events. So it's not a rare thing to have a furin cleavage site. See, the acquisition of polybasic cleavage sites by hemagglutinin has been observed after repeated passage in skill culture or through animals. So it can happen in passage in the lab, and it can also happen in passage in animals in the wild. So this Furin cleavage site does not point to one versus the other, just like the scientists that are kind of concerned about the lab leak theory say. Like you can get it naturally, or you can get it through serial passage in a lab. And then the other uh, one of the other things that people talk about is this specific CGG codon um, that is in the furin cleavage site, and people have made the claims that that is an extremely rare thing and it doesn't occur in any other coronavirus. But it's very clear that if you just look up MERS, this is MERS, Middle East Respiratory Syndrome. If you search for CGGCGG, well, there it is. It's right here. CGGCGG is right here, and it occurs in this and several other coronaviruses. So the statement that this sequence does not occur in any other coronavirus is just demonstrably false. Okay. All right. So that being said, let's go through this this quick Nature article that goes over some of these problems, some of these lab leak theories. All right. Scientists don't have enough evidence about the origins of SARS-CoV-2 to rule out the lab leak hypothesis or to prove that the virus has a natural origin. So right now, we don't have the information to say, did it escape from a lab or did it just make the natural jump? We do have enough information to say that the idea that it was genetically engineered is very, very unlikely. For it to be genetically engineered, a couple of things have to happen. One, someone has to have developed a technique that is not known and they would not have published it, that doesn't have a fingerprint that we can detect. So they would have to have done that. Then they would have had to use a sequence for the spike protein that is not known and is not similar to anything we've known before and was not predicted to work by computational analysis. Um, so those two things make it highly unlikely that it was genetically engineered. Not impossible, but highly unlikely. Now, did it accidentally leak from a lab? That is a possibility. Yeah, that's definitely a possibility. And so, but we don't have any information to say that one is better than the other. And from the information we have currently, I tend to think that the lab leak hypothesis is less likely. And there's a good paper that we're going to go over that shows why that is. And so, one of the things that they say is that we haven't found any of the animals that have the ancestral virus. And but what's known is that most of the emerging infectious diseases began with a spillover from nature. This was in HIV, a lot of influenza pandemics, swine flu, avian flu, Spanish flu. They were all came from natural jumps. Ebola, um, and so we know that these these come from bats generally because they're very similar. Okay. But still, the possibility remains that it escaped from a lab. Although lab leaks have never caused an epidemic, they have resulted in small outbreaks involving well-documented viruses. A relevant example happened in 2004, and they were studying SARS, and they got infected by SARS. So, what are the key arguments for a lab leak? One holds that it's suspicious that almost a year and a half into the pandemic, the ancestral virus has not been detected in people. I mean, in an animal. Okay. Is it suspicious that no animal has been identified in transmitting the virus to humans? Outbreak origin investigations take years and often culprits remain unknown. It took 14 years to nail down the origin of SARS epidemic. And then to date, so Ebola started in like 1976, and to this day, we have not found the um, the animal or the virus from the animal that started Ebola. So We so these things take a long time, and sometimes they're never found. Like Ebola still has not been found. I think HIV started probably in the late 1800s, and the animal source from a chimpanzee was not identified until like 1999. So that's like almost 100 years. And so here's proof of so remember SARS one started in 2002, and here's an article from 2017. So that is about 15 years later. That says Bat Cave solves mystery of deadly SARS virus and suggests new outbreaks could occur. And so it says after a detective hunt across China, researchers chasing the origin of the deadly SARS virus have finally found their smoking gun. In a remote cave in the Yunnan province, biologists have identified a single populated horseshoe bat that harbors virus strains with all the genetic building blocks of the one that jumped to humans in 2002. So again, it's from a cave in the Yunnan province. Yunnan province has a lot of bat caves, evidently. Um, and they found it, and this was 15 years later, so 2017. And then what's interesting here is at the end, it says another outstanding question is how did a virus, so SARS, the original SARS, the first cases were in the Guangdong province, which is 1,000 kilometers away from the Yunnan province. So it says another outstanding question is how is a virus from bats in Yunnan could travel to animals and humans 1,000 kilometers away in Guangdong without causing any suspected cases in Yunnan itself? So see, this is not unprecedented. People make a lot of they have a lot of problems with the fact that Wuhan, which is far away from Yunnan, was where the first cases of COVID-19. However, in 2002, when SARS came out, the first case was in Guangdong, which is also which is 1,000 kilometers away, a little bit closer, but it's 1,000 kilometers away from Yunnan, where the ancestral virus was found in the animals. And here in 2017, they say, although many markets selling animals have already been closed or restricted following outbreaks of SARS, Yuan agrees that the latest results that the risk is still present. It reinforces the notion that we should not disturb wildlife habitats and never put wild animals into markets. Respecting nature, he argues, is the way to stay away from the harm of emerging infections. Obviously, we did not take heed of this, and it's likely that this happened again. Okay, so it's not suspicious that we haven't found the ancestral virus from an animal yet, because we're a year and a half into this. It took 14 years to find SARS. We still haven't found Ebola, um, and it took almost 100 years to find HIV. And then, it's like Spanish flu. We never found it because, I mean, we don't have the technology to find the ancestral source. So that's not surprising. And it's also not unprecedented to have the index human case in a large city far away from where the ancestral virus is. Because in SARS, it happened in Guangdong, which is 1,000 kilometers rather away from Yunnan, where the ancestral virus was found in animals. All right. Does the virus have features that suggest it was created in a lab? And so we went over that. No, it does not. The features basically say that it was not engineered. All right. And then, this is talking a little bit about the CGG thing. About 3% of nucleotides in SARS-CoV-2 uh, contain CGG, but around 5% um, uh, of those encoding are gene the virus that caused the original SARS, so the first SARS, are CGG2. So the CGG motif is not uncommon. So people that are making a big deal out of that are, they're not looking at the data correctly. And then people think that it's very well adapted. However, it's not. It's just adapted enough. It infects a bunch of other animals, minks, cats, a bunch of other animals. It's not uniquely adapted to humans. Did research collect SARS-CoV-2 from a mine? Not that we know of. They collected that RAT kind of ancestral one that's like 96% similar from a lab, but not this one. Okay, but I mean, it is something that should be investigated, definitely. It's possible that it escaped from a lab. Very unlikely that it was engineered, but it's possible they were studying the thing in a lab and passaging it, and it did infect someone and it escaped from the lab. Okay, so if you're still with me and you want to know more, I'm going to go over this paper, um, which is The Origins of SARS-CoV-2, a critical review. There's a lot of authors from many different institutions across the world, Canada, um, Australia, the United States, London, New Zealand, China. So lots of different places. All right, so I'm going to just kind of read through this because it was a very interesting paper, and I'll link this paper in the description. And if you guys want to stay with me on this video, please do. All right. Since the first reports of a novel SARS-like coronavirus in December, there's been intense interest in understanding how it emerged. Recent debate has coalesced around two competing ideas: a laboratory escape scenario and a zoonotic emergence. Here we critically review the current scientific evidence. Okay, so evidence supporting a zoonotic origin. Coronaviruses have been long known to present pandemic risks. This is why the Wuhan Institute of Virology and lots of other labs study coronaviruses, because we've known for years, and they've been predicting for years, that coronaviruses are dangerous. They have high recombination events, and they are known to jump from animals to people on a regular basis. And then SARS-1 basically, and MERS was one of the more recent events of that. So SARS is the 9th documented coronavirus that infects humans and the 7th identified in the last 20 years. So this jump has been made 7 times in the last 20 years. Fortunately, the previous ones have been controlled either by non-pharmacologic methods and public health methods, or they turned out not to be that scary. Um, the emergence of SARS-CoV-2 bears several signatures of these prior zoonotic events. It displays clear similarities to SARS-CoV that spilled into humans in Guangdong province in November of 2002, and then again in Guangzhou in the Guangdong province in 2003. Both of these were associated with market selling live animals and involved species, especially civets and raccoon dogs, which were also sold in the Wuhan markets in 2019 and are known to be susceptible to SARS-CoV-2 infection. So civets and raccoon dogs are susceptible to this. We know that. Okay, so basically they're saying that this has happened before. It's not an uncommon event. Coronaviruses um, do infect animals, and they are infected in the animals that are sold in these wet markets. And SARS-CoV-2 does infect civets and raccoon dogs, which are also sold in these markets. SARS-CoV-2 is similar to 4 endemic coronaviruses. These coronaviruses are the coronaviruses that cause the common cold. It's most likely that many of us have been infected by one of these, and they're just kind of circulating around in the world all the time. This HCoV HKU1 also has a furin cleavage site. And then this is a map showing the index cases that we know about in humans and their location, their proximity to these wet markets and to the Wuhan Institute of Virology. So if you see, I mean, most of them are clustered around the wet markets and not that close to the Wuhan Institute of Virology. And these in red, these in the triangles are linked to the Hunan market, and the squares are linked to other markets, and the circles are linked to none of them. So most of them, the ones that we can have linkages, um, are linked to the Hunan market. So they think, based on epidemiologic data, that the Hunan market in Wuhan was where it started. But this is a lot of this information is circumstantial, obviously, because we haven't found the ancestral virus. But it may take years to find it, or we may never find it. Could it have escaped from a laboratory? There are precedents. In 1977, there was a pandemic that started from a large-scale vaccine trial challenge, where I think they used an attenuated vaccine that wasn't attenuated enough, and the vaccine itself caused the pandemic. Okay, so this is an important piece of information. So known laboratory outbreaks. So when the laboratory causes the outbreak, then you have people that worked at the lab or their family members as some of the index cases. That only makes sense, right? So what happens is, despite extensive contact tracing of early cases during the COVID-19 pandemic, there have been no reported cases related to the laboratory staff at the Wuhan Institute of Virology. So basically, none of the early cases had worked in the Wuhan Institute of Virology, and they tested the entire laboratory of Dr. Shi, who does the coronavirus research at the Wuhan Institute of Virology, and all of them had not been exposed to SARS-CoV-2. So that information makes it pretty unlikely that the Wuhan Institute of Virology was the source. Now, is it possible that China is lying about this data? Yes, it's possible. But if you believe the contact tracing, there none of them were related to the lab. Okay, considerable attention has been devoted to claims that SARS-CoV-2 was genetically engineered or adapted in cell culture or humanized to promote human transmission. Yet, since its emergence, SARS-CoV-2 has experienced repeated sweeps of mutations that have increased viral fitness. So what their argument here is that some of the people say, okay, so maybe it wasn't genetically engineered using one of these reverse genetic systems that have a fingerprint. However, you can passage the virus in human cells, and over time you kind of can breed the virus without using genetic techniques to be adaptive to humans. However, what they're saying is with all the variants that we've seen, they've had increased fitness. And why did these increased convergent mutations that increase fitness of the virus to people not come up immediately? So what they're basically saying is the virus was actually not optimized. Like this Delta virus that's out right now is way more optimized than the virus that showed up then. And also, um, SARS-CoV-2 is a generalist virus. It's capable of efficient transmission in multiple species, including minks, tigers, cats, gorillas, dogs, raccoon dogs, ferrets, and large documents have been documented in minks with spillback to humans. So basically, this virus isn't optimized for people. It just happens to infect people along with a bunch of other animals. And if it was passaged in people, it would probably more specific to people, is what they're saying. And then this Furin cleavage site is found in other viruses, so they don't find that to be very suspicious. So their conclusion is, as for the vast majority of human viruses, the most parsimonious explanation for the origin of SARS-CoV-2 is a zoonotic event. So they're basically saying almost all, like the vast majority of all previous pandemics have been zoonotic events. The documented epidemiological history of the virus is comparable to previous market-associated outbreaks. So they're saying that the spread and the contact tracing of the early cases are very similar to the original SARS. Um, and then the laboratory escape theory says that basically it came from the laboratory. That means people that worked at the laboratory should have been some of the index cases, but they are not. So they contend that there's a substantial body of scientific evidence supporting a zoonotic origin. While the possibility of a laboratory accident cannot be entirely dismissed, it may be nearly impossible to falsify. This conduit for emergence is highly unlikely relative to the numerous and repeated human-animal contacts that occur routinely in the wildlife trade. So what they're saying for the laboratory accident to happen, a lot of things have to line up, and it's a one-off event, and all the data doesn't point toward there. Like there were no all the index cases were associated with the market. None of them were associated with the Wuhan Institute of Virology. And then there are multiple contacts between humans and animals every day in the wildlife trade. So basically, they're saying that is a more likely scenario, although you can't rule out the laboratory leak scenario. Okay, so to sum this up, the three scenarios are engineered virus that was purposely released. that seems very, very unlikely based on the information that we know. Laboratory leak, accidental leak from the Wuhan Institute of Virology. That seems unlikely because of the index casing, which doesn't implicate the people at the Wuhan Institute of Virology and more implicates people that visited one of the wet markets. However, that thing is a much more likely scenario, and that definitely could have happened. And the zoonotic um, transfer, there, I mean, there's no evidence to support that over the others as well, but it just seems uh, it's like the default because that's most likely how it happened because that's how it happened in the past. So people are sticking that as a default until there's some strong evidence to point to the lab leak theory, which right now what little evidence we have points away from the lab leak theory. So again, no one knows what the source of the virus is. That's still under investigation. But basically, a lot of the things, such as like why is it in Wuhan instead of Yunnan? Um, why haven't we found the ancestral virus in the animal? These furin cleavage sites, the spike protein, is it adaptive to people? The CCG motif is rare, and the furin cleavage site is rare. All of these are not viable arguments to make based on the information that we have now. Now, if there's new information that comes out, that like evidently there was someone from the Wuhan Institute of Virology that was the index case, or we find out that there was a virus that they were storing in there that was almost identical to SARS-CoV-2, well, then that would totally change the calculus of this and make the lab leak theory more likely. But as of now, I think the lab leak theory is pretty unlikely. All right, thanks for watching.